I'm really very happy to moderate this webinar. My name is Moez Hanin. I'm an academic professor in life science and biotechnology. I'm also coordinator of several European projects in research and innovation. Today in our webinar, we will be dealing with multi-omics technologies, one of the powerful technologies today for different life science fields, especially in medical research, in particular in precise medicine. And it's really great pleasure to have with us today Dr. Tori Blackwell, who will be talking about this topic. And before really getting the subject at hand, I let you, Tori, present yourself briefly.
Yes, hello, can you hear me?
Yes, perfect.
Okay, perfect. Yes, it's my pleasure also to be on this webinar of a digital innovation partner. And yes, thanks for the kind introduction. I have been working with single cell technology since quite a while, so I'm happy to share what we have been working on, what the shape of the future of this technology might look like, and how we can deploy this for finding new drug targets.
Hey, thank you, Tori. Thank you for this short introduction. I think before really starting and sharing with you the agenda, just I would like to let you know that you will be recording this webinar. I hope this is fine with everyone. So as you can see, I will be introducing the Digital Innovation Academy, which is really hosting this webinar and organizing this webinar. And afterwards, I will give the floor to Tori to talk about single-cell multi-omics. He will be describing somehow the workflow and also focusing on single-cell RNA-seq analysis and providing you some few examples of applications. And at the end, you will get an idea about the boot camps dealing with this topic that we'll be developing and offering next year. And we will end up with a small discussion for any questions that you may have during this webinar. So let's get started very briefly to introduce you the Digital Innovation Academy. Digital Innovation Academy is really created by the Swiss Digital Network as one of the latest value propositions. And in few words, Swiss Digital Network is a hub of different entities which are specialized in IT technologies. These cells are working as independent but complementary cells in IT technologies. So they are offering consulting, engineering, but also training services. And in the frame of this training activities that we have developed the Digital Innovation Academy. So if you go to the next slide, you see that this Digital Innovation Academy was launched by the Swiss Digital Network. It has the main objective is really to democratize digital transformation key capabilities and help people to get the knowledge and succeed their digital transformation journey. So this Digital Innovation Academy is managed by one of the cells, Digital Innovation Partner, which is composed of experienced academic professors. We have senior engineers, data scientists, and they are all together in charge of developing and maintaining and delivering these training courses. And DIP is collaborating very closely with different universities, Swiss universities, but also other international universities, and also with experts from other cells who have also expertise in different IT technologies. Actually, the current portfolio is already quite rich, and it's offering different training activities covering different topics, like effective site reliability engineering, effective MLOps, machine learning for DevOps, but also machine learning for life science. And it's in the context of machine learning for life science that we are investing efforts today to develop new training boot camps dealing with single cell multi-omics. This is what we will be talking today about during this webinar. And I will now give the floor to Tori to tell you more about these technologies, and you will get at the end an idea about the bootcamp that we can be offering you next year. So Tori, the floor is yours.
Thanks, Samuel. It's great to be here, and I would like to start the webinar with Biologic 101. So to answer the question, what is actually a single cell before we dive into single cell technologies? So, and as you can see from this study that was conducted in 2023, so there's still a lot of research going on what is actually a cell type. But if we imagine the cells of our body, we have to understand also that each cell of one individual has the same DNA, and it's only the genes that are expressed in this in particular cells that are making the cell type enabling the cell type to perform its specific function. So for instance, in the heart, we then have the cardiomyocytes that help us to pump our hearts, or in the brain, we have some cells that make us understand the webinar or to look at the slides, for instance. And in this study, for instance, which you can also explore interactively on the website, we can see that there are different cell types. Overall, they studied 400 cell types, and that they differ in cell count. So if we take into account all the cells, we have like 36 trillion cells in our body, considering in this example a male of 70 kilo, but they also investigated this for children and female. And we see that, for instance, most of the cells, if we go by cell counts, are from white blood cells. But if we go by cell mass, it can be different, as there the most cells occur are skeletal myocytes. But if we take this apart, then we have a lot of adipocytes. The message from this is that we have different cell types that can different cell mass and cell count. And the only thing that makes them different is that gene expression program within a cell that then leads to different function. But the complexity that that we have to also to take into account that in a disease, various cell types might be affected differently, so that some expression programs might change there. And with single cell technology, we want to study this in depth. So, and actually single cell technology is the first method that enables us to study this gene expression program in individual cells. Before, it was not possible. Before, in research, we took like one tissue or one organ, and then we could look at the gene expression program from this organ or this tissue, but it was like an average of all the genes that are expressed throughout the tissue. But now with single cell RNA sequencing technology, it's really amazing that we can study this at single cell level. And as you can see from this plot, we have like a rising number of cells per publication. And this is a steady increase of cells that has been sequenced and where the information is available. And also that this correlates well with the number of cell types that has been reported. So this drastic increase of cells and nuclei samples will help us to understand more complex disease conditions, or to also improve to understand the heterogeneity that occur between different patients within a certain disease, or also to increase our understanding of different cellular aspects. For instance, if we include different modalities, meaning different measurements, or multiple organs, while classically maybe for one disease, one organ has been studied. And since we are confronted with a lot of data, the long-term perspective and also short-term perspective is to deploy artificial intelligence for medical breakthrough. And this is really where the field is moving right now. So therefore, it's very important that we understand both sides and that we connect this for the new innovations in life science and medical research. So because if we think about multi-omics, we can think about drug target development, testing the effectiveness of drug targets that have been already deployed, or also to identify new biomarkers for diagnosis of the patients. But one thing that we always start with is like a patient heterogeneity. This can be caused by genetics, as we all have different mutations. This can be caused by environmental effects, lifestyle, and other effects. So with multi-omics, we can then first of all get different angles for this co-funding factors, and also we can then capture the heterogeneity, because now we have the possibility to look at the single cell and capture the differences in patients that experience actually the same disease. And from there on, when integrating multi-omics together, we can then come to a hypothesis to test new drug targets and to develop new approaches for personalized medicine and biomarkers. So when we talk about data integration with single-cell multi-omics, every clinical data can be integrated. So for single-cell multi-omics, we can capture different modalities that I will explain on the next slide. But also we can incorporate diverse biological data sets, like for instance, genomics, phenotypic aspects, pathogenics, genetics, and other sources that might be linked to patients. So this can be combined also with single-cell multi-omics that are different aspects that you can learn from the single cell. And therefore, this provides a holistic approach for better understanding of disease mechanism. And for single-cell multi-omics, we have different technologies. And today we will mainly talk about single-cell RNA sequencing, but I just want to briefly show you what other technologies and other modalities are out there that could be integrated into a multi-model approach. So first of all, we have single-cell RNA sequencing, where we measure in a single cell the transcriptome, meaning like which genes are expressed in a single cell, so that it can fulfill the cell type specific functionality. But there's also single-cell attack sequencing, which is used to capture the epigenetic changes or properties of a cell, where we can measure the chromatin accessibility. Then also there's single-cell high-seq, where we can capture the conformation of the DNA, which can be linked also to certain diseases, as this can affect the regulatory effects on a certain gene. And if we want to study more in depth the transcriptional regulation and epigenetic factors that might be related to a certain disease, we can use technologies such as single-cell cut-in-one, which can capture histone modification or transcription factors that are associated with certain regions on the DNA and also regulate transcription. And then there's single-cell ADT sequencing, where we can measure the surface proteomics. This is really important also, for instance, to identify on a single-cell level proteins that we can find on the surface. This can be, for instance, important to design specific vehicles that can target the cell to deliver like a therapeuticum, or as other purposes that we can differentiate different cells based on the surface, so that we don't have to break the cells. Then there's also TCR and BCR sequencing on single cell level to understand the VDJ combination in immunology, DNA methylation, which changes on the DNA that can also affect the transcription, and single cell DNA sequencing, where we just look at the DNA itself. This is especially important for cancer, because there you can have different copy number variations and different mutations that can affect the patient outcome. And also, this is just to give you a brief overview, as there are many other methods coming up, like or that are already established, like CRISPR-Cas screening, where you can have like genetic editing in single cells and therefore do this in parallel in a lot of cells and compare the output, so the effects of one alteration to other genes, for instance. RNA metabolic labeling, where you can track like very fast changes in the transcriptome, but also there are arising methods such as single cell proteomics and metabolomics. And right now, everything is studied in single cells. So that means we have like a tissue that then is disrupted and we isolate the cells from there. But at this point, we are very happy to have the single cell information, but also we are losing the information which cells are actually close to each other and which could connect. And there's an arising technology called spatial transcriptomics, for instance, where we can then connect this also in a spatial manner, so that we know which cell types on a tissue are located close to each other and has the potential to interact. So that, for instance, in a disease, there's an interaction that we want to inhibit. We can directly design targets for this approach. So, but let me also explain in a very brief setting how single cell technology and single cell RNA sequencing is working. So once we have like a solution where we have disrupted our tissue into single cells, there's a technology where then we have like a bead emulsion where we have like a small compartment of oil where we have only one cell and one bead. And this bead has some barcodes with a very unique sequence that will later help us to identify that this bead was together with this cell in a particular compartment. Because once we have compartmentalized the beads and the cells into one compartment, we will lyse the cell in this compartment, and that means that all the transcripts, like all the genes that are expressed, are now free and they will be captured by these beads. And together with this fusion, this leads to the linking of the very unique sequence that we have on the barcode and the gene that is expressed by the cells. Because the genes that are expressed by the cell, they have all the same sequence since they are coming all from the same individual. So, but with this barcode that we add, we really have the single cell information, so that now in a cost-effective manner, we can pool this together and sequences. And this is required because then we know which genes we can find in each cell, because if we have the sequence, we know nowadays exactly from which gene it is coming. So we can just count how many sequences we have from each gene and then perform some analysis based on this information. So with a transcriptome of a cell, it's usually not fixed, but a dynamic process as well. So therefore, we can capture this also with a technology. But apart from this, we can also mainly derive the function of the cell type, and the cell stage in a first approach. But of course, also if then there are some alteration due to different influences, whether be it the biological system itself, something externally like a drug or some other stimuli, we can measure the response of each individual cells with this technology now. So, and if you are new to this technology, I just want to guide you through some first insights maybe on how to explore this data. So there are a lot of data sets already available, although it's still increasing. But for instance, here there's one website that you can check out with a QR code, which is the Tabula Sapiens, where they collected single cell data from several human organs that then are projected in this low dimensional space. And here every dot actually represents one single cell. But since now we know also from which organ they are coming, we can study like what are the genes that are set on or set off in certain organs or cell types from the whole human body. So I think it would be very helpful maybe to get into the topic, because then The next part will be about the single-cell RNA sequencing workflow, and then some research that we have done in order to approach downstream analysis of single-cell and multi-omics technology. So I think it would be very nice maybe to now explore together a little bit the data set here. I will maybe change the screen so that you can have a look at it simultaneously, so that we can look or get a feeling about the data. Also, if this is not new maybe to you, I hope that this is new to you. So here you can explore the different cell type composition of the by count or biomass, and also for male, female, or for your favorite cell type. So this you can find under human cell tree map, but we will focus now on the tabula sapiens up. So with the QR code, you will usually or you should land on this page. So I will just maybe show you the QR code once again, so that you can go there on your own pace. Otherwise, this is then the whole tabula sapiens atlas. And then if you want to for this we don't need any data science skill at this moment, so it's very interactive and intuitively, so you can check out, for instance, one specific gene. So this is a well-known gene that for endothelial cells, so we can look where this is expressed. So we just click on this drop here, which is their information that we now want to see the expression colored in for this plot. So we see that it's mainly expressed in this region. So if then, 1 second, I have to move. So if then we are interested in what kind of cell type, for instance, it has been captured, or what developmental stage or donor ID, we can also explore this. For instance, here we see that it's expressed in highly in endothelial cells of the artery, because this is a known endothelial cell marker for many years. This makes a lot of sense, and here we see that this is mostly different endothelial cells from different organs. And also, if you want to check out the different cell types, for instance, we can also color them by cell types. So I will just maybe give you a couple of minutes to explore this on yourself before we then continue with the presentation, just to get a feeling of the data, because then as the next step, we will talk about how to generate this kind of low-dimensional representation of the single-cell RNA sequencing data in general, and what we can do afterwards in order to find biomarker or drug targets or other approaches that we would be interested in. And if you have some questions, feel also free to ask them already. So, but maybe we will continue then at 12:32 with a webinar, so that you have a little bit of time to explore the data.
Or people also can use the chat if they have any question. They can also ask the question.
This can be about the presentation already, or it can also be specifically about the resource.
Yeah.
Or also about the opportunities that are offered by the Swiss Digital Network. Anything is welcome, I guess. But maybe also it will take some time to check this out and to get a little bit familiar. So this is also just for yourself. It's not to get like a feeling. There will be no questionnaire about it, or we will just give this at a hand to have some first insight maybe on on how this data look like. So we will continue in 2 minutes. So I think we can then resume with the single-cell RNA sequencing workflow. As you might have explored the tabula sapiens, for instance, there we have like 500,000 of cells, and in each cell, usually we have about 20,000 genes. So just to give you an idea of the dimension of the data. But usually if we start with a single cell workflow, we start with, of course, with the data. So this can be of multiple source, single cell RNA sequencing, site-seq for surface proteins or other modalities. So, but right now we will just focus on single-cell RNA sequencing to get an overview of what will be done. So first of all, there will be alignment in order to count the genes that we capture in our sample. And then there will be some quality control to check how well we can define single cells and how well is actually the quality of the data in each individual cell. And if there will be multiple data sets coming from different modalities or from different resources, could be, for instance, different clinics, we also need to do data integration in order to correct for batch effects. And then there will be clustering and cell type annotation to add some meaningful biological and medical aspects to the data, and then actually starts the fun and interesting part, which are the downstream analysis to study intracellular processes within the cell or intercellular connections between different cell types, so that we learn about the interaction and how this influences each cell, so that we can perturb or use this knowledge to develop medical approaches. So in general, this is the start of each experiment. If we don't have the data yet, that we need to design the experiment. So if you start there, this is very important to take into consideration and to explore all the options. Also, if I mean, right now we will just go very briefly through it, as it is not the major part of this webinar. But also, if you need some help there, we are happy also to help you with experimental planning, or if you have some questions. But yes, so usually you need to decide which modality you want to capture, how to set up which protocol you want to use in the laboratory, and then once this is done, this can be sent for sequencing, where we obtain the sequence of from the cellular barcode that I showed earlier, and the sequence of the gene that is expressed together, so that then we can align it to the reference genome, because there we know the sequence from each gene. And since it's linked also to our cellular barcode, we can then count for each individual cell the transcriptome, so that we can come up with what we call a raw count matrix, which is like the 20,000 genes by X cells that we are analyzing in this experiment. And also for the workflow, there have been several frameworks established. For instance, single cell experiment for and Seurat for R, or ScanPy for Python. So they are very good to work with, and we can also store additional information there. For instance, metadata that are coming from the clinics that are associated with a patient, or other data that are linked to our samples. And when we choose a framework, it's also important to maybe consider what we want to do downstream, because some of the package actually require like a certain framework to be used upfront. So, but this we will also come later to. So then once we have done that, we need to do some quality control. There are some, in all of the packages, there are some matrices to assess the quality of our data on different levels. So whether we have enough gene counts, for instance, or actually the cell is in a healthy state, so that we can proceed with the analysis. Then we need, or the next step, what is performed, is to normalize the data to account for variability in the capturing of actually our genes, and doing sequencing. And then we can, since we have our data matrix, which contains like 20,000 genes and X amounts of cells, then we usually perform like a reduction of the data by projecting the high dimensional data into a low dimensional space. Here, for instance, what we saw also on the Tabula Sapiens is then a UMAP, where we can then reduce the dimensionality to have a better visualization for the data. And if we have data from multiple sources, also we need to correct for batch effects, because there can be maybe different conditions, different sample preparations, different sequencing platforms, different clinics that all process the data a little bit different. So we need to account for this, but especially, of course, if there are different conditions coming from different project partners, it's very important that we are not chasing some batch effects, but real biological or medical signals. But at the same time, also a challenge in batch correction is that we maintain the biological viability, so that we are still can capture the heterogeneity within the cells and between the patients, because that is something that we still want to explore later on. And if we start from scratch, then also we do some clustering, for instance, using the Leiden algorithm to identify distinct cell types that also have like a biological meaning. And once this has been established, and the cell types you can either annotate with the help of an expert that is that knows the tissue or the sample very well, so that it can help to identify some markers. But nowadays, also since the field has moved on, there are also like a lot of databases that can be used to annotate the cells and nuclei automatically. And then we can start with further analysis, for instance, differential expression, where we can check for gene expression programs, like genes that are switched on in a disease state versus a control state, or also between different cell types on how to see how they react to certain stimuli, for instance. And from here, actually, we can do a lot of downstream analysis that then require like a dedicated approach, but this can be, for instance, to explore further the cell type cluster. And I will just give you an overview here and follow up with some examples that we have done. But just to give you an idea about the possibilities, and also there are more and more approaches being developed. So this is growing as we speak. But basically, we can investigate the cell type cluster, differential expression programs to check which gene expression program changes in disease versus normal conditions. Pathway analysis. So pathways are like a set of genes that is grouped together because it fulfills like a biological process. So we can by also then grouping the genes together, we can account for this, or there's also trajectory analysis where we can have like a projection of the cells that is representing the time, for instance, in disease progression or the response to a certain drug. Then gene regulatory networks in order to understand the key players of a certain response, cell-cell communication, where we can analyze the factors that are expressed in the cell that are responsible for the communication with each other. So that, for instance, in one cell, there's one gene that is expressed that is well known to communicate with another gene of another cell. And this can also result in some abnormalities in disease, for instance. So then a strategy could be to inhibit or to disrupt this communication as a very simplified theoretical approach, let's say. So this is a kind of analysis that we can do downstream. So I will just guide you through examples of analysis that we have done, or tools that we have at hand on the different levels. So let's start with the genomic level, where we just look at the genomic profile of individual patients. Because each human being has like a unique sequence of their genome, which leads to small alterations in the transcriptomics, and this we can also capture on single-cell RNA sequencing level. And for that, we have developed a method that enables us to do high throughput single-cell RNA sequencing by pooling different patients together. And then later, based on the SNP profile, so the genetic profile, we can tell them apart, and we can identify also SNPs that are specific to one or the other patient. So this reduces the cost of single-cell RNA sequencing dramatically, and also helps us to understand the genetic background of certain patients. Then a classical approach is always to understand, of course, which gene expression programs are upregulated or downregulated, meaning switched on or switched off during a disease or in a certain cell type. And with single-cell RNA sequencing, we now also have the possibility to do this in a cell type and disease-specific manner. So, for instance, in this analysis that will be published also very soon, so this paper is accepted in Nature Cardiovascular Research, we can identify a new biomarker. For instance, here we compared two different types of endothelial cells that we defined in this study, and then on the vertical axis, we can then identify markers that are either higher expressed in this type of endothelial cells or in this type of endothelial cells, but also we can identify markers that are disease specific. So in this range, then we identify markers that are disease specific, but are upregulated in both endothelial cell type. But if you go in this part of the diagram, we can also identify markers that are disease specific and specific to this cell type, and the same here. So this enables us to find new biomarker and drug targets for novel therapeutical approaches, and also this can be used then to study more like the medical and biological functions. And And as I said, also something that is very important to study or very interesting and important to study is like the cellular crosstalk. So it is for some genes, it is known that they are expressed, for instance, on the surface and that they can interact with other proteins from other cells. So then there will be an exchange of information between the cells. And if we know what kind of information is being exchanged between the cells, this helps us to understand like how cells are reacting. For instance, also this doesn't have to be another cell. It can also be a drug that is addressing such a protein that is expressed on the surface. And to understand like how then the gene expression program within that cell is changed. For instance, then we see an upregulation or downregulation of a certain gene set, and also then we can use this to understand the system better, but also to therapeutically target this approach or to improve immune system response by its modulation. And this is actually also from the same study that I just showed you before, where now we looked at this interaction. But here, as I said, in single-cell RNA sequencing, we have single cells that are disrupted from the tissue. So here we know that these cells could potentially interact, but we didn't know whether they actually would be in close proximity. So therefore, in order to show you us a little bit the power of a multimodal approach, is what we then did is like to combine this with spatial transcriptomics, where we have the information where each cell is located in the tissue. So here, since we were studying atherosclerosis, this is tissue from the arteries, where then we can really pin down some so here these are known some of the interactions that could have been in the plot from before, for instance, where then we wanted to find out whether actually the interactions that we have identified there based on prior knowledge, whether we could also find them in close proximity so that there would be an actual interaction. Because if they are very far apart, it's less likely that there will be some communication. But here actually, so if you maybe remember, we identified like two types of endothelial cells, and we could also then identify like different intercellular crosstalk in different niches. So for instance, if we look at this interaction, we see that this is at the outside of the artery. So this is an artery, this is the outside, this is the inner part where the blood is normally flowing, and we see that this interaction is in proximity, but in most of the places, but it's not really specific to a place. But also we identified then, for instance, some interaction that are more located at the outside of the artery or the inside of the artery. And with that, we can use this knowledge then to directly develop drug targets or biomarkers for the microenvironment and this specific cellular crosstalk. And this we can do in an unbiased manner because like with a single cell technology, we are capturing all the transcripts that are possible, and while previously people had to design and think about the genes that they would like to analyze. But with single-cell RNA sequencing technology and also spatial, we are doing this in an unbiased manner. So in summary, I hope that I could convince you about single-cell multi-omics, and that the data in single-cell multi-omics is continuously increasing. So and that the integration helps to understand complex medical problems, and that this could reveal new possibilities for therapeutic development, and also that new bioinformatical approaches arise to shape the future of precision medicine, but also that there are a lot of things that are still open, and where new, especially AI approaches, are definitely needed for the next medical breakthrough or target discovery in the disease of your interest. And so this webinar hopefully gave you a good overview of all the different technology and data analysis that are possible within the single cell multi-omics world, and that can be then drive to clinical applications. But if you want to learn more and dive deeper into one specific topic, there will be some offering of boot camps where you can learn how to interpret single cell multi-omics data sets to obtain a clearer view of different cell types and the different technologies, as well as to dive into the interpretation of single cell multi-omics involving your focus. So kind of, but maybe started with the tabula sapiens today, but this will be really about specific data and really working hands-on on some data. So we will learn the basic steps of data analysis for single-cell multi-omics to evaluate public data or your own data sets, and there will be several boot camps that cover different entrance levels depending on your prior knowledge. So the audience that we are addressing with this boot camps are medical doctors and researchers, biologists, data analysts, or those that want to join the community of single cell multi-omics. And the only prerequisite that this requires is like curiosity to learn more about single cell multi-omics. Maybe also you can bring some questions to address, but this is not a pre-requirement. And for some of the sessions, there will be also hands-on in data analysis. Because in the first course that we have here, this will be just or not. This will be an overview of the technologies and the growth potential for biotech, but it will be without any data analysis or hands-on per se, but we will rather work on the interpretation and potential of different multi-omics that I presented earlier on. So not only single-cell RNA sequencing, but also CRISPR screens, for instance, or proteins or epigenetics. Then the second course is designed to do some data science, to get some introduction into data analysis for single-cell transcriptomics, so to analyze your own data set or public available data set, for instance, to mine this for drug targets or biomarkers, or to develop your own analysis and sorts on it. So this will be hands-on. We will be working in R and Python to do some actual work. And then the third course will be more advanced. So this will be following the second course, where then we also work on a downstream analysis of single-cell multi-omics. So if you are interested in these courses or have some questions, we are happy to receive them now, or you can reach out to the digital innovation partner for registration and further inquiries. And I think now we have the
Thank
interactive.
you.
Yes, thank you.
Thank
Q&A
you,
session.
Tori. Yes. Thank you very much, Tori. Thank you. It was really, thank you for your talk, a very exciting talk, and this insightful information, especially on the single cell RNA-seq analysis, which is really a very powerful tool to address complex human diseases. I think people can get an idea where we can go with such technologies. You gave also an idea about the bootcamp that we could be offering at different levels. And I hope people now they can if they would like to interact with us, they can ask any question. They can raise their hand, or they can put their question in the chat. And so you have also, if any question may arise a bit later, you can also send by email your question, and the experts will be ready to reply to you. Any questions?
Otherwise, I mean, it's also
Yes, maybe people
uh,
also need time also to integrate and
yes, this was
to,
probably a lot of information to
yeah,
digest.
of
Also,
course, yes.
the work that I mentioned, the publications, if this is something that you are interested in, I think so. They will be published soon. If you want, you can follow us on LinkedIn, and then also
Yeah,
you will get notified
absolutely.
immediately. And also, if you are just interested
Yeah,
in keeping the contact,
exactly. Yes,
please feel
please
free
feel
to reach
free.
out at
Yes,
any time.
yeah,
Yeah.
yes. Please free to reach out by email, and I hope this webinar was really interesting for you, and we really look forward to getting your feedback and comments, how we can help you and how we can guide you if you are really interested in such technologies. Thanks again for you also, Tori, for this very nice talk.
It was a pleasure.
Uh, I think we can stay in touch, and anyway, we have the email addresses of participants, and then we can keep in touch. If there is no questions, I think we are ready to close the session. Thank you all for joining us. Thank you for your time. I know it's not easy to reach out just during lunchtime. I hope you enjoy the webinar, and we wish you a nice afternoon. So thanks a lot, and hope to see you soon. Goodbye.